They're some of the most dynamic, dazzling, jaw-dropping displays in nature.
It's these rocket ships and explosions of sparks and spewing of what looks like blue smoke. And these long chains look like Japanese lanterns extending off into the distance as far as you can see. It's magic.
Imagine you are outside on a cool summer evening and you look up to see a flash of light fly by. What could it be? You look closer and there are a bunch more flying around. They are fireflies. But have you ever wondered, how does an insect make light? Well, the process is called bioluminescence, and that is what I'll be talking about today. What exactly is bioluminescence, besides the fact that it makes fireflies light up? The prefix bio means life in Greek, the suffix lumen translates to light in Latin, and the definition is the light emitted by organisms. Bioluminescence is also a special type of chemiluminescence, which is the term that describes the chemical production of light. Bioluminescence is more specific as it pertains to the process of living things producing light, since light from a light bulb is much different. However, each organism has a different way to light up, meaning most luminescent animals or organisms have their own specialized light-producing organ and their own light-producing cells, which are host symbiotic luminescent bacteria. Although there is a variation across different species, the chemical reaction that produces light can be simplified to one general process. Typically, a pigment called luciferin in the presence of oxygen is oxidized in a reaction that is catalyzed by an enzyme called luciferase. The oxidation of luciferin producing oxyluciferin releases energy in the form of visible light. Cofactors such as calcium and ATP aid luciferase in catalyzing the reaction of luciferin with oxygen. Sometimes luciferin, a catalyzing protein, and a cofactor form a single unit called a photoprotein, which can be triggered to produce light when a particular ion, typically calcium, is added to the system. In these cases, light is produced in flashes rather than a continuous flow. This occurs in some marine organisms through a photoprotein called aquaron, which is used primarily by the jellyfish Aquaron victoria. Not only fireflies and jellyfish luminesce, but many other organisms as well. Here is a Caribbean ostracod female anglerfish. This is the male Caribbean ostracod, and it uses its self-produced light to attract mates, while the anglerfish uses its bulb filled with luminescent bacteria to lure prey. And of course, there are a lot more than just that. But why would an organism need to light up? Well, in addition to attracting mates and luring prey, it can also be used for communication, protection, and illumination for organisms living in the pitch dark in the deep sea. The picture here shows how luminescence can be used to startle or confuse a predator. As I mentioned before, each organism has a different luminescent system. Some of the major systems include bacterial, dinoflagellate, Vargula, celocentrazine, and firefly. Although there is a large group of organisms that could be focused on, luminescent bacteria and fireflies are among the most common and well-known, so I'll be going into those in more detail. Let's start off with bacteria. Bacteria are the most widely distributed light-emitting organisms on Earth. Most are capable of living free, but the majority are found associated symbiosis with hosts. Bacteria are nourished with readily available food sources for growth, and the hosts utilize the bacteria in various ways. For example, pinecone fish use luminous bacteria in their ventral cavity to illuminate surroundings and for interspecies communication. There are three major genera: Photobacterium, Vibrio, and Photorhabdus. These different growing conditions and the reaction kinetics of luciferase, but all use highly homogeneous biochemical machinery to produce light. The biochemical process of bacterial bioluminescence involves a heterodimer bacteria luciferase with two different polypeptides, which are referred to as alpha and beta, or Lux A and Lux B. The active site of the enzyme is within the alpha subunit, and the substrates consist of the luminaire flavin mononucleotide, also known as FMNH2, molecular oxygen, and long-chain fatty aldehyde. Excess energy from the oxidation of flavin mononucleotide and the aldehyde, which occurs along with the reduction of oxygen, results in the emission of blue-green light at the wavelength of approximately 490 nanometers. Although luminous bacteria have the capability to produce light, not all of them do. Many bacteria express bioluminescence only under certain minimum population conditions. A fascination form of gene regulation called quorum. Sensing this phenomenon is by marine luminescent bacteria that live free in the ocean do not emit light, while those living in the localized or combined environment do. The catalyst in bioluminescence that induce the expression of the LuxCBABE gene are regulatory proteins and a small chemical compound called an autoinducer. Bacteria need a confined, nutrient-rich environment to accumulate autoinducers. Then, when the concentration of autoinducers reach a certain level, the luminous system is activated. By this system, bacteria sense when a significant concentration has been reached based on autoinducer accumulation, so that bioluminescence only occurs when nutrients are sufficient and the environment is favorable. Although bacteria are among the most seemingly simple organisms, they have an efficient system of both life production and gene regulation that allows them to use bioluminescence advantageously for their survival. I know this has been a lot to take in, so here's a fun fact. Bioluminescence produces a blue or greenish light because red light is the first in the light spectrum to be absorbed in water, so it doesn't travel as far. Next up is dinoflagellate. Dinoflagellate luciferin is derived from chlorophyll and has a very similar structure. At a pH of 8, the molecule is protected from the luciferase by a luciferin binding protein, but when the pH lowers to around 6, the free luciferin reacts and light is produced. A modified form of this luciferin is also found in herbivorous Usagi shrimp, which could be a dietary link for the acquisition of luciferin. I will be showing you guys an example of a dinoflagellate algae later on in class. Varguline or Cyprindia type luciferin is found in the ostracod, also known as sea shrimp, and is also used by the midshipman fish Porkythes. Here there is a clear dietary link with fish losing their ability to luminesce until they are fed with luciferin-bearing food. It has been shown that ostracods synthesize this molecule from the amino acids tryptophan, isoleucine, and arginine. Colinterazine is the most popular of the marine luciferins, found in the variety of phyla. This molecule can occur in luciferin-luciferase systems and is famous for being the light emitter of the photoprotein acurin. Although they are not as common as bacteria, fireflies are among the most widely observed with bioluminescent organisms. Fireflies are able to control their light by controlling when they add oxygen to their light organs. When oxygen enters the light organ and binds to the cell's mitochondria, the presence of nitric oxide allows the oxygen to flow into the light organ to set off the reaction. Because nitric oxide breaks down quickly, the flashes can happen with the high frequency. There are a variety of reasons that fireflies light up. Larvae display their glow to communicate their bad taste, while adults have flash patterns that are used to identify members of other species and members of the opposite sex, and in some cases for mating. Fireflies produce light by chemical reaction in their light organ in which ATP, firefly luciferin, and firefly luciferase are combined. Firefly luciferin is an organic compound with a ring system and the chemical formula C11HAN2O3S2. The luciferin is oxidized, which releases energy in the form of visible light. This results in brief flashes in the abdomen of the firefly. The light emitted varies from green with a maximum wavelength of 535 nanometers to red with a maximum wavelength of 630 nanometers. This variation is due to different forms of oxyluciferin forming at different acidities, with red being produced at a pH of 6 and green at a pH of 8. The generally accepted mechanism involves a process in which the D-firefly luciferin reacts with ATP and magnesium ions to form lucifero-adenylate. A proton is taken from the C4 carbon of the adenylate by a side chain amino acid of luciferase. A molecular oxygen is added to the anion and the electrically excited oxyluciferin molecule and carbon dioxide are formed. Red light emission occurs from the keto form of the emitter or the oxyluciferin at the pH of 6 as previously mentioned, while yellow-green light emission occurs from the adenylate anion of the oxyluciferin at a pH of 8. The light emitted by fireflies is cold light without a lot of energy being lost as heat. Humans also have some uses for bioluminescence. Bioluminescence has primarily been used in scientific research, specifically through fluorescent tags. This is when a fluorescent molecule selectively binds to a specific region or functional group on the target molecule. Common tags include lithium bromine and fluorescent proteins. Through the use of fluorescent detection of specific cells and living organisms can be accomplished, such as in the brain where fluorescent probes have been used to reveal the mechanisms of neuron communication and the activity of surface receptors on brain cells. In addition, by using fluorescent tags, the mechanisms of the infection process of several types of viruses can be determined, particularly in how the virus enters the host cell. Luciferin and luciferase have also been used in biological research, space exploration, and oceanography to detect traces of ATP. The possibility of using bioluminescence as a light source has been considered, and there are lanterns made from polarized fireflies. However, generally bioluminescent light is too dim to provide more than an ambient glow, and though attempts have been made to make bioluminescent bacteria brighter, there have been little success. Since these organisms sounded so cool, I wanted to see them in real life. I decided to go to the Monterey Bay Aquarium. Bioluminescence is both fascinating and visually amazing. It's a show of the incredible power of organisms that adapt to various situations in the world of biochemistry beyond the production of light. Bioluminescence captures both the beauty and complexity of the natural world, from attracting prey and hiding from predators to communication and reproduction. The production of light has become much more than the release of energy, but rather a component in an intricate web of interspecies and interspecies or relationships. Although it is not directly connected to humans, it has provided us with the means for making discoveries in science, particularly in the field of biology, and acts as a possible resource for lighting. Thus, bioluminescence, though it may primarily be in microscopic organisms, tiny bugs, and creatures miles and miles under the sea, it's a critical component to humans as well as a stunning insight into the living world. Thanks for watching, and I hope you enjoyed learning about the chemistry of bioluminescence.